Hello everybody, welcome to the YouTube channel of MARATHA VIDYA PRASARAK SAMAJ KTHM College Nasik. I am Dr. Jayashri Bandal from Department of Microbiology KTHM College Nasik. We are here to study the paper Enzymology of the TYBSc class semester 1. The contents of the course are as per the SPPU syllabus. We have already started studying the chapter 1 titled Enzymes, and now we are studying the topic 2 of the chapter 1, which is Role of Riboflavin as a cofactor in metabolism. In this lecture, we are going to study the history of the cofactor, the occurrence of riboflavin in various natural foods, its structure, its role in metabolism, the causes and symptoms of deficiency, and the recommended dietary allowance. To introduce the cofactor or the vitamin, riboflavin is one of the 8B vitamins essential for human health. It is found in foods and is also available as a dietary supplement. Enzyme cofactor, it is as an enzyme cofactor, it is present in many flavoprotein enzyme reactions. It is a water soluble vitamin, and we are going to see the disadvantages of being a water soluble vitamin. A brief history of the discovery of this particular vitamin. It was Alexander Blyth in 1872 who observed that riboflavin is a yellow colored pigment in milk showing yellow fluorescence. It was Richard Kuhn who actually has given a lot to the discovery of the vitamin and its role in human health. So it was Richard Kuhn along with Paul Carrer in 1930s who characterized the riboflavin, showed its chemical structure along with some other scientists like Zent Gorgi and Wagner Janerjee in 1933. For this and for some other contributions also, they were awarded a Nobel Prize in 1937 and 38. It was Kuhn who first proved that riboflavin is an essential growth factor. Along with Mervyn, Kuhn and his team first synthesized riboflavin in 1930s. Earlier, riboflavin was synthesized chemically in the laboratories, but nowadays, with the Ashbia gossypi and Bacillus subtilis, with these microorganisms, it is exclusively produced as a product of microbial fermentations. The natural sources of vitamins include green leafy vegetables, sprouts, almonds, milk and milk products like cheese, whole grains, that is unpolished grains, and the animal products like chicken, eggs, and animal liver. Commercially, as I told in the earlier slide, the vitamin is produced by using the yeast, bacteria, and by microbial fermentation process. Riboflavin is a vitamin which is made up of two components, the ribitol, which is derived from the sugar ribose, and a flavin component. It is a component of the coenzymes flavin adenine dinucleotide and flavin mononucleotide FMN. The FAD and FMN are tightly bound to the protein part of the enzyme, and so they are bound as prosthetic groups. FAD and FMN participate in the redox reactions during the metabolism. They work as coenzymes for about 150 redox reactions that are taking place in a cell. The structure of riboflavin, as indicated earlier, consists of a sugar ribitol and it consists of the isoalloxyzine ring, which is called as flavin. This justifies its name riboflavin because it contains the sugar ribitol derived from ribose and the flavin component, which consists of the isoalexosine ring. In this isoalexosine ring, the nitrogen atoms at the position 1 and at the position 5 are the ones which are actually playing a role in the redox reactions. So it is the isoalexosine ring which is important in the biochemical reactions to follow. So riboflavin is consists of a three-ring structure, that is the isoalexosines. Here you can see the three rings which is linked to the ribitol moiety. It is yellow to orange colored, it is in powdered form, and it is soluble in water. The active forms of the coenzyme forms of riboflavin, as mentioned earlier, are flavin mononucleotide (FMN) and flavin adenine dinucleotide (FAD). So the structure of flavin mononucleotide is, here you can see the isoalexosine ring, the ribitol, and the phosphate. So it is flavin mononucleotide. Why nucleotide? Because it resembles a nucleotide which contains of a nitrogenous base. Here this is somewhat considered similar to the nitrogen base. There is a sugar moiety and there is a phosphate, and these are the three components of a nucleotide. So the FAD and FMN, these coenzyme forms of riboflavin, are also called as the pseudonucleotides. This is FAD. So FAD, as the name suggests, flavin adenine dinucleotide. So there are two nucleotide-like structures. So you can see one here and you can see one here. So this is the FMN. This is what we have seen in the structure above. So this is the isoalexosine ring, this is ribitol, and this is phosphate, which resembles a nucleotide structure. Whereas this is our adenine mononucleotide AMP. So you have adenine, you have ribose, and you have a phosphate. So this is a pseudonucleotide bound to a nucleotide. So there are two nucleotides. Thus it acquires the name flavin adenine dinucleotide. To illustrate the role of riboflavin or its biochemical function, riboflavin in its coenzyme forms has the potential to undergo oxidation reduction reactions. So FMN or FAD both can bind to two hydrogen atoms, converting them to FMNH2 or FADH2, the reduced forms of the coenzyme. The coenzyme forms are involved in major reactions that are the major metabolic pathways that are taking place in the cell, like metabolism of carbohydrates, proteins, and fats. We are going to exemplify this with some few reactions later on. Activation of vitamin B12 and folate, protection of erythrocytes and other cells from oxidative stress. Now, the enzymes that use flavin or riboflavin as the coenzymes are called as flavoproteins. And those flavoproteins which contain metals are called as metalloflavoproteins. The metals commonly engaged or bound to these flavoproteins are molybdenum and iron. So, coming down to the biochemical function, as I said earlier, it is the isoalexosine ring, the three-ring structure, which is reduced to form the FMNH2 and FADH2. So here we can see that this nitrogen at the fifth position and this nitrogen at the first position are the ones that are reduced. So this is how they bind to two hydrogen atoms and they get reduced. So the oxidized forms are yellow in color, whereas the reduced forms are colorless in nature. In between, there is one more step where a semi-reduced form is formed. So both these hydrogen atoms don't bind together. Sometimes it is one hydrogen which binds first, forms a semi-reduced form, and then the second one binds to this to completely reduce the coenzyme form. Now we come down to the examples in the various pathways or the various metabolic reactions that are taking place in the cell. The first one that is very important is the lactate dehydrogenase, which catalyzes the conversion of lactate to pyruvate. So here you can see the lactate with the help of FMN, which will carry or which will accept the two hydrogen atoms from lactate, converting lactate to pyruvate, and the FMN gets reduced. The next example is amino acid oxidase, which converts the amino acid to keto acid. This is a reaction which is very important in protein metabolism, whereby the keto acid then can enter the central energy yielding pathways. In this reaction, ammonia is released and FAD is reduced to FADH2. So here you have an amino acid. This is the typical structure of amino acid. With the help of FAD and oxygen, it gets converted to a keto acid. And here you can see ammonia is released and FADH2 is formed. In the metabolism of carbohydrates, FAD is really very important, and this is something that we should understand very properly and very meticulously, because most of our energy we are getting from the carbohydrates that we are consuming in our diet. And all of us know that carbohydrates form a major part of our diet in the form of rice or rotis and all this. So, the pyruvate dehydrogenase complex, which allows the pyruvate to enter the TCA cycle by converting it to acetyl-CoA. This pyruvate dehydrogenase complex is a multi-enzyme complex which consists of three enzymes, namely pyruvate dehydrogenase, dihydrolipomide S-acetyltransferase, and dihydrolipomide dehydrogenase, of which the dihydrolipomide dehydrogenase enzyme requires or uses FAD as the cofactor. Pyruvate dehydrogenase complex, as I said earlier, is involved in the oxidative decarboxylation of pyruvate to acetyl-CoA. And this reaction is not as simple as it appears here. You've already studied the working of pyruvate dehydrogenase complex last year. So FAD, as I said earlier, is a component of one of the three enzymes that constitute the pyruvate dehydrogenase complex, and which is dihydrolipoyl dehydrogenase. So here you can see that pyruvate along with the reduced form of coenzyme A and water is converted to acetyl-CoA and CO2 is released. The second enzyme, which plays an important role in the metabolism of carbohydrates in the TCA cycle, is the succinate dehydrogenase, which converts succinate to fumarate. And in this reaction, you can see that the H2 from here are removed and FAD is converted to the reduced form FADH2. The third important enzyme which participates in the metabolism of carbohydrates is alpha ketoglutarate dehydrogenase, which converts alpha ketoglutarate to succinyl CoA. So FAD is a part of dihydrolipoyl dehydrogenase complex of alpha ketodehydrogenase, also like the PDH complex. So this enzyme converts or catalyzes the conversion of alpha ketoglutarate to succinyl CoA, wherein the FAD is converted or reduced to FADH2. In the metabolism of lipids also, the FAD plays an important role as a cofactor of the enzymes involved in the metabolism of lipids. So acyl-CoA dehydrogenase is an important enzyme in the metabolism of lipids, which catalyzes the conversion of fatty acyl-CoA to 2,3 unsaturated acyl-CoA. So the fatty acyl-CoA is converted to 2,3 unsaturated acyl-CoA, wherein the FAD is reduced to FADH2. In the metabolism of purines also, during the excretory metabolism, FAD plays an important role as a prosthetic group or cofactor of xanthine oxidase, which converts xanthine to uric acid, which then needs to be excreted. So, understanding the metabolic role, you now know that FAD or in turn riboflavin has to be a very important part of our diet because in absence of FAD, in deficiency of FAD, we now can say that the metabolism of proteins, the metabolism of carbohydrates, and the metabolism of lipids is going to be affected. And these are the major energy generating nutrients that we are consuming. And so, obviously, the energy generating process will be affected. We'll have less amount of energy, and that is what will lead to the fatigue that we might experience due to the deficiency as an overall symptom. The causes of deficiency are, since it is water soluble, it is continuously excreted, and this demands the regular intake of the vitamin in our daily diet. The deficiency can be classified as the primary deficiency, which is the deficiency in diet. We are not consuming the recommended amount of riboflavin in our daily diet. And the secondary deficiency can be due to your defective intestinal absorption in spite of consuming the recommended amount of the vitamin in the diet, or increase in the excretion, something wrong with the excretory mechanism or the processes related thereof, and so an excess amount of the vitamin is excreted from the body, or the vitamin is absorbed but something wrong with its assimilation or its use, and so ultimately the body is not able to use the vitamin at the right places. The deficiency symptoms can be classified as the oral symptoms, then a general symptom leading to anemia, facial and ocular. We are going to see each one in detail. Now for the anemia, the vitamin is included in RBC production and it transports oxygen to the cells, and so it leads to anemia. It also is seen to alter the iron absorption, which is a leading cause of anemia. So, coming down to the details of the oral symptoms that are seen to the deficiency of the vitamin, which can be called as glossitis or termed as glossitis and angular cheilitis. In glossitis, the mouth and tongue are inflamed. That is, you get ulcers in the mouth and tongue, reddening of the tongue, ulcers in the tongue, or the whole mouth, the oral cavity is due to the deficiency of riboflavin. The angular cheilitis is your corners of the mouth or corners of the lips. They develop cracks, they become dry. In facial symptoms, you have dermatitis, that is, you can see dry and scaling skin or white patches on the skin. And it also leads to a certain scale. Due to the scaling of the skin, it leads to dandruff and seborrheic dermatitis. The ocular symptoms include cornea vascularization, that is, bleeding in the cornea. So the eyes appear to be deep red, reddening of the eyes, very frequent reddening of the eyes, and the eyes become very sensitive to bright light. Bright light cannot be tolerated, and so the eyes keep on adjusting themselves to the bright light, and so we can see a person blinking his eyes very often. The recommended dietary allowance uh, is can be defined as the estimated amount of the nutrient, that is, in this case it is riboflavin per day, which is considered necessary for the maintenance of good health. So the recommended dietary allowance for riboflavin is for adults, it should be 1.2 milligrams per day in our daily diet every day. I've also told you the natural sources which contain the riboflavin. So we need to be keen in including those natural sources of riboflavin in our daily diet to maintain the daily intake of 1.2 milligrams in our diet. During pregnancy, this requirement is increased, so you need to increase the consumption of riboflavin in during pregnancy, so it has increased to 1.4 milligrams per day. During lactation, a feeding mother also has to increase the amount of riboflavin in her daily diet. Now, to summarize what we have studied in this lecture today, I think all of us have been very sure now that riboflavin is quite needed for growth and overall good health. It helps the body to break down the carbohydrates, proteins, and fats, and thus it is involved in a number of reactions in the body that are required to produce energy, that is ATP. And due to the lack of energy, obviously, we are going to be fatigued, we are going to be tired due to the lack of ATPs released. It is important for the health of our eyes also. We have just seen that in the earlier slides. The diet rich in riboflavin has been found to lower the possibility or the frequency of uh, developing cataracts. It is important for pregnancy health and a healthy fetus development because there are many processes that are involved in the development which need riboflavin as its coenzyme. It has also been observed that the incidences of severe migraine they decrease almost by 50% by taking higher doses of riboflavin. By saying higher doses, I mean to say that the doses higher than the recommended dietary allowances that we have seen in the early slide. It definitely improves memory because your overall energy level in the body is dependent upon the metabolism of all these basic nutrients and production of energy. Riboflavin, along with vitamin C, it facilitates to increase the level of glutathione, that is the antioxidant. And so it helps to reduce the oxidative stress and it is very important for us to know that oxidative stress is one of an important reason for the development of tumors. So riboflavin plays its small role to help reduce the probability of development of tumors by reducing the oxidative stress with the byproduction of or increasing the level of glutathione. So, thank you very much, and I'm very sure you have understood the biochemical role of that FAD and FMN or the coenzyme forms of riboflavin plays in our metabolism. Since it is involved in so many pathways, the recommended dietary allowance of this vitamin is slightly higher than that of the other vitamins. And so I think all of you all will pledge now to include the natural dietary resources in your daily diet every day in the recommended allowance from today. Thank you very much.